Hi everyone, thanks for coming for today's Volume Imaging Australia webinar. I'm really pleased to have Dr. Yunfeng Hua here with us to share his insights on protocol development and osmium-based staining mechanisms for high-throughput volume electron microscopy. And he's going to talk about the history of staining protocol development, things that he's learned along the way, and what they've been doing recently in rodent organ preparation for X-ray microscopy and volume EM. He's leading a research group at Shanghai Institute of Precision Medicine with a focus on auditory neural circuits. And yeah, I'm really excited to pass over to you and hear everything you have to say, Yunfeng. Thanks for joining us.
Okay, thanks for the introduction. So it's a recording, right? Okay, so maybe I need to pay attention to what I say. I thought that it was a very informal discussion, but yeah, I mean, I prepared some slides, mainly from my GRC talk, but with more detail. As you will see, the title of today's talk will be Progress in Volume EM and Sample Preparation, and maybe a little bit more. Okay. There will be my talk will focus on two parts. One is prepare large biological samples for the volume EM. The second is plan efficient acquisition of volume EM. So, okay, and for this audience, probably I need to emphasize too much about electron microscopy. So as you know, for the TEM, we can image ultrastructure of, for example, synapses, and also with scanning electron microscopy, we can also map the sample surface to image the topology or even 3D structure from top layer. But to get the 3D information, the conventional way a long time ago, what we do is we try to cut the slice. Yeah, we try to cut the sample into its consecutive slices and image one after another. And for example, 1 micron, people need to cut like 10 or 20 slices and image them. Therefore, by definition, our volume EM is okay. The sample thickness larger than 1 micron. The technique we can use is called volume EM. For me, I would rather classify volume EM in two different arts. One is serial block-based art. So with this machine, we can cut the sample surface with a diamond knife or with a focused ion beam and image the newly exposed surface in an iterative fashion. And the other way is we can also cut the sample and collect or fish the slices onto a grid or even a Kapton tape so that we can image afterwards, use TEM or use scanning electron microscopy. Okay, but for both technique, actually I benefit from M-bloc EM staining. So for example, this is a sample within a resin block. Before we can image, for example, use the serial block phase EM, the sample need to be heavily stained and also resin embedded. And for that, we need to have sufficient conductivity. Otherwise, this incident beam, electron beam will cause surface accumulation of electrons that you won't be able to image anymore. And also, it has to be very high contrasted, and also that reduce your draw time and also reduce the radiation damage so that you can cut properly. And for the other type of microscope, serial sectioning art, so actually it's recommended because in principle, they can cut the slice and then do a post-staining. But if you have a perfect sample, they don't need to do the post-staining anymore, and then that will reduce the chance of the slice to be damaged, and also to avoid staining heterogeneity, and it can also accelerate the imaging. Okay, so let's go through the protocol development. It is a long story. If you look back in the literature, so in the earlier in the 60s, so the osmium amplification with TCH has been developed. So actually, one round of osmium stain is followed by carbon kazajide plus the second round of osmium, so that you can amplify the osmium signal by at least one-fold. And also the reduced osmium is also introduced by mixed osmium with ferrocyanide, so that you can, with this staining buffer, you can stain the sample quite nicely. So until 1984, so actually people combine these two methods and then use on cell culture. They have very nice contrasted structures under EM, so that these methods is the gold standard back to that time. Okay, I think the first one who used the LOTO protocol for brain sample is one in Mark Elisman's lab, although this is a conference paper. And then 2 years later, Jeff Leishman's paper has published a proper or a landmark paper to describe this method. And also 2010 or 2012, it's the time volume EM become more people pay more attention to this technique because of the connectomics. Okay, so actually that's also the time when I joined the lab. I joined Moritz Helmstaedter's lab to do volume EM. So initially, I was quite naive. So I thought, okay, so use the protocol, we can do large sample. And the first trial we do is we take a 1 millimeter brain biopsy punch and then stain the sample with the LOTO protocol and then cut them into half and then image the block face to check, okay, if there's anything we can improve or to check the staining quality. And immediately we realized that, okay, this is not easy. So as you can see, this is the block face we imaged, and the outer ring has a very nice contrast, nice structure. But in the intermediate areas, you have a lot of precipitate. And even you can't see the structure very clearly. And in the core region, you will have less contrasted structures, and then often you have soft resin in there. So this pattern actually is very disappointing because that tells us no matter how long you prolong the incubation, you will always have this pattern. It's not due to insufficient incubation time. There's something wrong or produced by osmium mechanics. Therefore, we try to solve that, but luckily, actually, with simple separation of osmium staining and also incubation and the ferrocyanide incubation, it gets solved. I mean, it's some kind of luck, but later on we found out, okay, this is pure chemistry. So it's in between you need to find a balance point between the penetration and the reducing and binding. Meanwhile, there's also some other technique has been introduced. For example, from Mikula et al., they use furamide to stabilize reduced osmium and then replace TCH with pyrrole for prolonged incubation so that it can also prepare very large samples. And also from Kevin Bergman's lab, people use extracellular brain space, extracellular space preservation protocol so that they can also push the LOTO protocol to a larger volume. And as you probably noticed that, I mean, these three labs, they are hardcore cerebral phase lab. I mean, you can imagine back to that time how we are desperate to push the sample size to a large volume because that defines how large we can image with the cerebral phase EM. And nowadays, there's more progress going on. For example, my colleague in Helmstedt's lab, they follow the sequential osmium and the reduction protocol at lower temperature and adopt the pyrrole amplification. They can even do whole mouse brain sample preparation. And also for the other lab, from Jeff's lab, they simply follow the 2015 protocol with preserved ECS so that they can prepare large clinical samples. Actually, for these kind of sample, you cannot perfuse of course, you cannot perfuse the patient, right? So usually from the surgical biopsy punch or from the surgical dissecting, they collect those specimens for imaging fixation. Okay, maybe after some time, people might consider combine these two methods with ECS preservation and also with this low temperature EM staining. Okay, so it works, but in terms of a mechanism, it's still difficult to understand. I mean, even for now. So there's a lot of chemistry behind that. So in our earlier paper, actually, we proposed some of these chemical reactions, and then recently has been reviewed by people from Chicago. They have made minor changes. I don't want to explain all this complicated chemistry. I rather simplify the key process. So one is osmium-8. So it is osmium tetroxide can penetrate through the membrane bilayers quite easily by alternating or by changing configurations. So second, when the osmium try to stain the sample or stain the membrane, so actually need to be reduced to osmium-6, and then osmium-6 will enter in the water phase by hydrolysis, and afterwards it further reduced to osmium-4. The osmium-4 has a black color. Usually we call the osmium dioxide. Actually, it has a higher affinity in the organic phase. It like to deposit on membrane bilayers. So with a very simple test tube experiment, we show that, okay, so after the staining, the osmium black always in the organic phase. And also in this paper, they did some chemical analysis for that to prove, okay, osmium black osmium 4 is really the contrasting agent for membrane staining. And the third is, okay, so osmium can be reduced in the membrane by unsaturated membrane lipids. That can also be reduced by ferrocyanide or other chemicals to osmium-6, so that you can bypass the slow reduction within the membrane bilayer, but rather can push the osmium-6 be produced and then maximize the staining of or production of osmium-4. Okay, these are the key points. With this, actually, we can explain a lot of phenomena we observe during our staining. For example, for the osmium, a simple osmium staining, we will have a very low efficiency because all the osmium will penetrate through the sample, and then before they can be reduced, if you do a washing step, it can be washed out quite easily. Therefore, in the end, you will have reduced or quite a weak staining of osmium. And then for the reduced osmium, so it causes penetration barrier. So because all the osmium will be in the reduced form, and then once they enter in the membrane, it's difficult to get out. Therefore, it form a kind of barrier for the further round of osmium penetration. Therefore, you have a very heavily stained surface, and then the core region is very weakly stained. And that is our understanding of reduced osmium. And then in our protocol, actually, we call this a Hua protocol, but actually it's named after, not by us. So we call it a sequential osmium penetration ferrocyanide reduction protocol. But it's a very long name. And I think people from other institute is nickname is Hua protocol, but we simply follow that. Actually, for that, we just let the osmium penetrate through the sample for the first round, and then use the ferrocyanide to then reduce the osmium so that you can lock all the osmium in the membrane bilayer. Actually, that's the logic behind that. Okay, so far, we back to like 10 years ago, it's already 10 years. Okay, we saw that, okay, it's the logic, it explain everything, but people become greedy and want an even bigger sample. And then in this paper, actually, they put the test tube or staining tube in the X-ray microscope to monitor the staining process. And surprisingly, they found, over a long time, the reason why they do that is because they want to determine, okay, how many hours they need to stain a bigger sample, bigger than 1 millimeter, right? So actually they need to measure the diffusion constant and also how this fixation and the staining can take place. And what they found is in prolonged incubation, in the presence of ferrocyanide, the osmium has been washed out gradually. So actually that's contradicted to what we proposed like 10 years ago. And even more clear, so in another paper, so for example, our Hua protocol works for 3 hours, and if you prolong the incubation, you will have a sample with a weakly stained outer ring, and then actually it's heavily stained core. I mean, that's the inverse contrast. I mean, of course, we need to update the model a little bit more. So a simple explanation for that. So I was thinking about it for a while. A simple explanation for that is, okay, so the osmium incubation is fine. So the ferrocyanide reduction probably is also right. And it works for short incubation at room temperature. But at for longer prolonged incubation, so as you know, those chemicals they cause some high osmolarity, or even the osmium can cause overfixation. So the membrane structure won't be protected or won't be intact afterwards after this prolonged incubation. Therefore, you have a lot of membrane breakage, and then also the material will be extracted from the sample. Therefore, if you don't have any structure anymore, or the osmium even is reduced and then previously stained the membrane bilayer, it will be washed out. And this is also observed in this kind of sample. The membrane structure is terrible, and then you will also have a lot of extraction from the cellular structure. And then simple solution for that is you do everything on ice, or maybe at a lower temperature. Actually, this is what we observed. So we simply put the brain sample, a small piece of brain sample, in CAC buffer or even in phosphate buffer, and then at a different temperature, either at 4 degree or at room temperature. As you can see, after 3 hours, so I mean, more or less okay. But after 24 hours or 2 days or even 3 days, and you will see the test tube is completely black, and also your sample slowly will be dissolved in this kind of buffer. But if you staying at a lower temperature, usually the sample will still be intact, and then can be further processed in the next round of, for example, ferrocyanide or TCH amplification or some other steps. Okay, so I think with that, people are quite happy, and my colleagues in Beijing, they try to work on more organs, so not even brain. Actually, what they do is they can stain lung, kidney, heart. In principle, all kinds of these mouse organs can be properly stained with a simple protocol. You don't need to modify anything. Just the incubation or the staining time is quite long. I remember several weeks. But in principle, in the end, you have very nicely stained sample, and then they do CT scan, they do EM, block phase imaging. As you can see, all the membrane structure, mitochondria, and yeah, even the yeah. So I don't know all these kinds of organs, but it looks fine to me. So this is a large sample preparation has several advantages. So one is it allows a large-scale multimodal imaging. For example, you can do CT scanning, afterwards you do EM scanning, or even you can prepare for design your imaging procedure or session. And another thing is you can do organ level mapping of diverse or rare cell types. Oftentimes in such organ, so you want to capture a certain cell type at different developmental or even in the different cell cycle stage. So therefore, prepare a large sample will be very helpful. And also, for the clinical users, they can archive a lot of pathology samples. For example, if you have a cancer tissue, Either you can do RNA-seq, you can also preserve and take a small piece. When we talk small, it's like a millimeter-sized piece of tissue, and then you can stain and embed them in resin. So those resin embedded sample they can keep for years or even centuries, you know. And whenever you are interested in the ultrastructure, you can go back and then cut some slice at an image. I think for us, it's very helpful to archive those rare or precious pathological samples. Okay, this is something I want to share with you about the large biological sample preparation. In principle, it's a solved question, so no more challenge anymore, at least for us. We will be happy to share with you the detailed protocols. I promise, Nicola, like a month ago, I want to put on bioRxiv, but thing got delayed. We are still testing different buffer combination, and because we don't want to use CAC buffer, it's highly toxic. We want to find a replacement for that, and we still want to do something to make that easier. And the second part, actually, I was quite excited to share with you. It's plan efficient acquisition of VEM. I think for most of the VEM users, extremely long data acquisition is a big problem, and also also very costly. Okay, the first dilemma we encountered is isotropic resolution and large field of view. So if you have a FIB, right, you have super fast, super high resolution in XYZ, up to 4 nanometer, I remember, but the field of view is quite limited. But if you have a cerebrophase EM with diamond knife, usually you have limited Z resolution, but the cutting is fast, and then the block face is bigger. As you can see, you need to make some trade-off between these two type of machines. Therefore, our initial idea is there any way we can improve the cerebral phase EM towards at least make the sample, the data quality as good, not as good as FIB, at least close to FIB. Okay, the second dilemma you might think about is when you plan a VM acquisition, right? You need to think about, okay, what's the sufficient voxel size you need to use for reconstruction? I think for neuralized reconstruction, it's like 15 or even 10 nanometer. And then you need to think about, okay, what's the optimal signal-to-noise ratio you need to put on each images, right? And also this sums up is the reasonable costs. So how long you do want to spend like a month to acquire one data set, as I do actually. I used to in the early stage of my career or my team development, I spent months to collect one data set. So I mean, the current dogma is the higher signal-to-noise ratio, the better. So actually, I believe that. I mean, I don't know if you believe or not. So, but if that is true, actually, we just want to simply test that. So we use a public FIB data set of volume size with a voxel size of 5 nanometer, and we downsample it gradually to 10 and to 15. And then we use a re-registration either by taking out these slices every 2 or every 3, or taking out these slices, or we add synthetic noises to these blocks. And then we use a so-called effective spatial resolution to quantify or to assess this data quality after all this manipulation. So actually, this is something we use for fluorescence microscope. It's called it's a 3D Fourier transform. So you don't need to remember the name, but it's a very simple. So in principle, you just need to convert your sample into Fourier space and then calculate. Okay, so when the resolution drop below the 50% of signal-to-noise ratio, you define this as your effective spatial resolution. Okay, what we found is, okay, so in that direction is we adding more and more noise, in that direction as we taking out more and more slices. Okay, what you will find is at very high Z resolution, or if we Z sampling rate, so the noise impact is very small. So actually, no matter how fast you image the volume or the image, so actually the data quality maintain the same, but only at low Z resolution. So actually the noise effect kicks in. So you will see the noise will make your data quality worse in an accelerated fashion. So that tells us, so effective resolution is more robust for data of high Z sampling. So which means if you, in Z, if you cut more slices, then actually you can keep a data quality in a nice optimum like optimum region. So if you have a worse Z resolution, then you need to image your sample with high signal-to-noise ratio. This is something we learn from this kind of modeling work. So therefore, invest in signal cutting rather than slow imaging is the message we got. Okay, then we think about, okay, if the scaling speed is not crucial, if there's any way by machine learning we can accelerate the VEM acquisition. So the initial idea is that, okay, so we use a slow scan, like a 2 microsecond per pixel slow scan to take a template image with a low noise. And then we don't need to take every image like that. So afterwards, we take like snapshots, like 0.5 microsecond per pixel. Of course, you have a lot of noise, but in this case, so we just need to provide a little bit of information, and afterwards, and then we can validate the structure again with a slow scan or reference. So this is the result we have. As you can see, the bright image followed by 3 noise images. This is AI denoised. In principle, can remove all the noises from the data set. It's even better than the ground truth. So that tells us, okay, at least for VEM, not every images you need to take as nice as possible. Then we want to do these things very properly. Then this is our imaging session. So with Cerebroffice EM, we acquire 2 datasets simultaneously. One is slow scan, served as ground truth. So we cut 50 nanometer, 300 more slices, and then image them at 2 microsecond dwell time. And then the other snapshot stack, actually we just trigger the acquisition afterwards. So actually these 2 images, they are identical. They are taken from the same block phase, but rather at a very fast speed, like 0.5 microsecond. Then we can synthetically generate a data set. So we're taking out the intermediate slice, so every 2 slices, and then use AI to interpolate the missing slice. Or we can use the reference slice combined with those snapshot slices, and then to mix the slices so that we can use our denoising algorithm to denoise this stack. So therefore, we have this a combination of data sets to validate all the AI methods. So this is the result. As you can see, this is ground truth. Of course, all the structure has been nicely resolved with the white arrow. And then in the snapshot images, of course, many features you can't see anymore. Therefore, we put a red arrow. So this is 2D denoise. So we don't use 3D information. We only use AI-based 2D denoising. Of course, many structure will be preserved, nicely preserved, but there are a lot of artifacts in there. And then this is the AI-based interpolation. So of course, there are some structure, for example, here the mitochondria, right? If you compare to the raw data, the mitochondria somehow gets wrong predicted due to the continuity, so between 2 slices. And of course, in our case, the 3D denoising, we call it the volumetric denoising. We want to differentiate distinguish us to those like temporal serial denoising. They call it 3D denoising. We also call it volumetric denoising. Of course, you will have some artifact. It's not perfect, but in principle, we can preserve more detail based on the ground truth. So actually, what we learned from this AI-based imaging processing, first is volumetric denoising is in general is better than 2D denoising. So which means the 3D context does contribute to our denoising, right? Because anyways, it's a structure in 3D, right? It's continuous structure, so you can easily define where is the noise, which is discontinuous in 3D space. So, but all the membrane, mitochondria, all these larger structures, actually, they are continuous in 3D space. And the second is, it seems like the reference frame is not contribute that much to our denoising. So, right, the reference frame is not essential for denoising if you compare these two cases. So, in this data set, we have this reference frame, but in this case, we have the reference frame, but in this case, we do not have the reference frame. But in the end, the outcome are very similar. The third is actually the volumetric denoising is better than the Z interpolation. Of course, the noise frame is always better than missing frame, right? In this case, the computer need to guess what is in between two slices, right? Usually this is generative methods. It cause a lot of problem in terms of artifacts. Okay, this is the message. What we learn from AI is totally different than our intuition or our human visual perception. Usually people like nice images, but in principle for computers, computer only care about the structure if it's continuous in 3D space, right? If you see the structure in the previous slice, you see the structure in the current slice, you will see the structure in the later slice. Actually, that's all the information computer needs. It's different than what we need. Okay, it's also very good for the cerebral phase users, because if you can image faster, then you can not only reduce the dwell time, you can also reduce the radiation damage. That means you can cut the sample with diameter knife even thinner. So then you can improve the Z alignment. Then again, data quality will be improved. Okay, then we try to build a self-supervised, because in this case, we don't have any reference anymore. We build a self-supervised AI to do the denoising. As you can see in the raw data, you can't see any structure anymore, at least for me. But after denoising, all the structure has been nicely restored. And also in the YZ, right? So, I mean, this is 20 nanometer cuts with this cerebral phase EM. I think probably this is the current record for this type of machine. But in principle, you can see that the resize also very nicely reconstructed or nicely imaged. So, of course, we did the same quantification for that. As you can see, the data set quality improves if you cut thinner, and then after denoising, so actually it can be pushed to a very low level. So the lower the resolution, the better the data quality, right? So actually in the denoised data, the 50 nanometer can be reduced by half to 25 nanometer, and then further pushed to 20 nanometer. And then more nice thing is if you plot the effective resolution against the cutting thickness, as you can see, actually it's a nice linear fit, suggesting, okay, so the resolution is only influenced by the cutting thickness, so the noise influence is minimal. So in principle, we can reduce all the residual noise. Okay, that's more or less I want to share with you today. Maybe some take-home message. It's pretty long. So first thing is prepare large biological sample for VEM. So the long incubation plus low temperature solves everything, more or less up to 3 days, I would say, if you want to ask me for the detail. And then it works for all the mammalian tissue types. So maybe for insects, for plants, maybe it's different. But for mammalian tissue types, this can be used in general. And the second part is plan efficient VEM acquisition. Thin cutting rather than fine imaging is preferred. So always think about, okay, so the resolution play even a crucial role in the VEM data quality assessment or data quality for the analysis. And the second is volumetric denoising using AI is possible. So we can use 3D context to do the 3D denoising. And the third is tune acquisition for AI rather than human. So usually AI or the machine has a larger memory. They can remember like hundreds of slices, not like human. We can remember less slices, right? And they can do low contrast comparison. For example, it's digitalized. You don't need to auto contrast, right, for machine. So inside the machine, they only compare numbers. So therefore, actually, it can do very low contrast comparison. They have a very wide range of perception. So as long as it can capture the images, they can do a lot of powerful things. Okay, so that's the end. I would like to thank my team. It's a rather small team, and, including Fang Wang, Jiang Yi, and Wen Jing. Actually, this is the core team for the data analysis and data acquisition. Hao Yu is our IT specialist to take care of the large data processing, and I have two more students. And I have a lot of collaborations, including Han in Beijing. Actually, it's a very close collaboration, and they have done this large sample preparation, and also together we have pushed the volumetric denoising things, and also other groups we collaborate to solve biological questions. Yeah, that's all. Thank you for your attention. I'm ready to take questions.